Hello, I'm Mun Su Kwon from South Korea, and today I'll be talking about my research shortly, a machine learning framework for extracting and structuring biological pathway knowledge from scientific literature. Biological systems are inherently complex, involving metabolic, regulatory, and signaling networks, and these networks often appear in the literature as static pathway diagrams. So, understanding such networks and their associated large-scale data is a critical challenge in modern biotechnology. So our goal is to turn these images into structured and machine-readable data. For this, we used four major biological and chemical databases. PMC is an archive providing a vast amount of bioliterature. Metaseq, KEGG, and Metanex offer information on metabolic pathways and enzymes. Lastly, we also use bio-based chemicals map, which compiles validated pathways for hundreds of industrially useful chemicals. So based on these backgrounds, here is a question. How can you extract structured information from pathway images in large-scale literature? To answer this, we developed a machine learning framework called eBPI. For the development of eBPI, we first curated a high-quality data set. We collected over 2,300 of images from 5 different databases. Also, we use data augmentation to generate additional samples for detecting errors in general. To capture both text and errors, we collected 100 pathway images from 89 unique articles. Additionally, we extracted nearly 59,000 terms from Metaseq database related to genes, proteins, and metabolites. These are used for both training and evaluation. And here is a brief overview of eBPI. It detects arrow position using FastRCNN, the object detection model, and extracts text via OCR and integrates them to identify biochemical reactions. This is an example of a biosynthetic pathway of gamma hydroxybutyric acid from Wikipedia, and this was not in our training data set. And eBPI was able to extract and structure biochemical reactions in a tabular format successfully. Next, we applied eBPI to large-scale literature from PMC. We selected 466 target chemicals from bio-based chemicals map and collected articles with the keyword metabolic pathway. And we extracted images, then filtered out non-pathway images using a very lightweight, efficient net model. The left figure shows the examples of non-pathway images like graphs and microscope photos. And using EfficientNet, we filtered the initial 670,000 of images down to around 50,000 pathway images. Using those pathway images and eBPI, we discovered target chemical specific reactions for hydroxytyrosol, phthalate, levulinic acid, 2-methylbutyric acid, and 1,4-butanediol. These biochemical reactions were not registered in KEGG, and in some cases not in Metanex either. So this highlights eBPI's potential to complement existing databases. In summary, we developed eBPI, a machine learning framework to detect biochemical reactions from pathway images, and it combines faster RCNN, OCR, and large-scale literature mining to generate new insights and extend current databases. I'd like to thank Professor Hyunwoo Kim and all lab members listed here, especially Jungu Lee, the co-author of this work. Thank you very much for your attention.